Hi everyone, this is part 2 of the June 2023 paper 2 biology A-level past paper. This is the syllabus Biology B. So part 1 covered questions 1 and 5, and now part 2 covers the rest of the questions. Starting off with question 6. The diagram shows an action potential in a neuron. Part A1, which of the following causes a change in potential between X and Y? Alright, so what's happening from X to Y is that the membrane potential is becoming more positive, and that is due to sodium ions coming into the neuron. So that is C. The reason why this happens is because you have these voltage-gated sodium ion channels, and they open, and that allows for the facilitated diffusion of sodium ions. Alright, let's move on to part 2. Explain why the membrane potential caused the change in potential between X and Z. So X and Z is referring to this part, which is the repolarization. So the reason why the membrane potential becomes more negative is because we have these things called voltage-gated potassium ion channels, and they open, and so that allows the potassium ions to diffuse out of the neuron. Alright, and then the other mark is at the same time, the voltage-gated sodium ion channels, which open to allow this to happen, are now closed. And that would be the 3 marks. Part B, the photograph shows an African crested rat. This rat has several adaptations to prevent it being eaten by predators. These adaptations include the rat chews the bark of a tree called the arrow tree, and the bark of this tree contains a poison called obane. So that would be an example of an anatomical adaptation, because there's a change in the physical feature of the rat. Statement number 2, the rat rubs the poison onto its fur, which is hollow, to absorb the poison. So the rat rubbing the poison onto its fur, that's behavioral. And then the third statement is the rat produces proteins in its saliva that prevents the obane from harming it. So that would be physiological. So physiological refers to changes in the processes that happen in the organism. So the answer for this would be B. Let me write that down. All 3 statements. All right, part 2. Obane inhibits sodium-potassium exchange pumps. The diagram shows the effect of obane, so it inhibits that first. So the diagram shows the effect of obane on the potential difference across the motor neuron membrane. Explain how obane prevents the transmission of nerve impulses. So the role of the sodium-potassium pump is to maintain the membrane potential. So what it does is that it pumps 3 sodium ions out and pumps 2 potassium ions into the neuron. And that causes the neuron to maintain its negative membrane potential. So this is what I would write in the text. All right, so this is what I would write. When the pump gets inhibited, you would have less sodium ions that are pumped out and less potassium ions that are pumped in. So that would be 1 mark. Let me just zoom in a little bit. Okay. So for the second mark, you would say that the resting membrane potential is not maintained, right? And because it becomes more and more positive, we can see that on the graph, that causes the concentration gradient for sodium ions to become less steep. So this is a second mark. The less steep would be a 3rd mark. And then because you need a concentration gradient for diffusion to happen, if you don't have it, then diffusion does not take place. That would be the 4th mark. All right, let's move on to question 7. Scientists investigated the role of ex situ conservation of plant species on genetic biodiversity. The scientists investigated the variability of DNA sequences of 30 different genes for a number of species of plants in the seed banks and in the wild. Part A1, which row gives correct statements about the genetic code in eukaryotes? So the answer for this would be D. That's because number one, it is not overlapping. We know that the codons are read in threes. So it's read like 1, 2, 3, first, second, third codon, fourth, fifth, sixth codon, seventh, eighth, ninth codon, and so on. So it's read in threes. The second point is it is degenerate. So that means that multiple codons can code for the same amino acid. Right. And lastly, they do contain introns, which, if you recall, do get spliced out during post-transcriptional modification. All right, part 2. Explain why the DNA sequences of genes are more variable than the sequences of amino acids in the proteins coded for these genes. All right, so this is what I wrote. First, the genetic code is degenerate, meaning that the 3 bases can code for a single amino acid. So that's the first mark. The second mark is it contains introns, and introns can vary. All right, part 2. Describe how the scientists can use technology to find out whether the gene sequences of 2 species are similar. There's actually 2 ways to go about this, but I've mentioned one of the ways. So the scientists can sequence the DNA. They can sequence the DNA of the 2 species and then do DNA hybridization. So DNA hybridization is a technique that we can use that aligns the 2 sequences together and that looks for similarities and differences between the 2 sequences. Another method that you can use is using gel electrophoresis. We know that if we use gel electrophoresis, it can produce patterns of bands, and we can compare the patterns of bands and see, is there more similarity or are there more differences? All right, so that is part 3 done. Part B. The scientists measured the mean genetic diversity of species in the wild and the mean genetic diversity of the same species in seed banks. The species examined include trees, non-woody species of plants, plants that are classed as endangered and not endangered, and plants of economic importance that are harvested by people. The scientists also measured the mean number of differences in the DNA sequences of the seed that has been stored for different lengths of time compared with plants in the wild. Okay, so here we have the graph. The y-axis is the genetic diversity, and then the x-axis are the different species of plants. And here we have the error bars, and the error bars represent the standard deviation. If they overlap, then that means that there is no significant difference between those 2 bars. Here we have another graph. The y-axis is the mean number of differences in DNA sequences from seed bank compared with wild plant, and then the x-axis is the age of seeds. Okay. Analyze the data to discuss the results of this investigation. So the way to go about approaching these kinds of questions is to comment on 3 things. First of all, describe the data. So we have 2 graphs here. So you would describe what are the trends you see. Are there any statistical significance between them? And the second thing you're supposed to do is explain why you see what you see. So for example, you see a trend here. Why would it be like this? And you would explain using what you learned in school and the information that they've given to you about this data. And then lastly, you can comment on the validity of this data. Is the sample size large enough? Are there differences? Are the differences statistically significant or not? So that's how you go about approaching it. And this is the answer. We're just going to zoom out so we can go through it together. So the first thing I spoke about was the description of the data. So if you look here, if you compare the non-woody species and the non-endangered species and the trees, we can see that the genetic diversity is greater in the wild compared to the seed banks. So that's what I wrote here first. There's a higher genetic diversity when conserved in the wild. And these error bars do not overlap. So that's commenting on the validity of the data. And then I would write out an explanation. Well, why would that be the case? Because in the wild, they have the chance to sexually reproduce. and sexual reproduction results in more genetic variation in the offspring. So going back to the same graph, I also mentioned that there is a difference between the genetic diversity in the wild and in the seed bank for trees, but in the wild it's just slightly higher. And this is what I wrote. So there is genetic diversity between trees in the wild and in the seed bank, but it is quite small. Okay, and that's probably due to trees having a longer lifespan. That does not give them enough time to increase in their genetic variation. Okay, so another thing I also commented on was the endangered species. So the endangered species is the only group in which there is a high genetic diversity in the seed bank compared to the wild. So then this is what I wrote. On the other hand, it's it's the opposite for endangered species, where it has less genetic diversity. That could be because there is a small population and there would have been more inbreeding, and you'd have a smaller gene pool as a result. Okay, I also mentioned how for species of economic importance, there is a difference, but that difference is not statistically significant because the error bars are overlapping. Okay, so that's what I wrote here. Now, I also commented on the second graph, as you can see here. So the first thing I did was comment on the trend. So we can see that as the age of seeds in the seed bank increases, the mean number of differences in DNA sequences also increases. And I wrote that there was a positive correlation, but that positive correlation would be a little weak. And then also another explanation for what we see in the graph. So I wrote down that this correlation is seen because as time passes, the species in the wild may experience selection pressures. Sorry, that looks like I'm crossing it out. Might be experiencing selection pressures, and because of that, they would accumulate changes in their DNA, which can cause them to become a little bit different from the species that are in the seed banks. Sorry, let's scroll back up, because the whole point of doing this is to investigate the role of ex situ conservation. So that would be conservation in seed banks, right? So that was the whole purpose of doing that. My final sentence was that because there are much greater differences as the age increases, it's important to keep updating the seed bank and keep rebreeding so that high genetic variation is maintained. All right, so that is question 7. Let's move on to question 8. Explain why a double circulatory system enables mammals to have a high metabolic rate. So the term double circulatory system just means that it has to go through the heart twice. So for mammals, our heart pumps blood to our lungs, and then from the lungs it goes back to the heart, and then from the heart it goes to the rest of the body, and then goes back to the heart. Whereas a single circulatory system, like a fish, it would only pass through the heart once in order to complete a circuit. So whenever we talk about the circulatory system, it's always important to mention that we're delivering oxygen for respiration. So I wrote maximize the amount of oxygen for respiration. So that would be 1 mark. And how does it do that? Well, it ensures that high blood pressure is maintained, right? That's why after the lungs, it goes back to the heart. And then from the heart, it goes to the rest of the body. To be able to deliver the blood to the lungs and the rest of the body. Okay, so that's the 2 marks. Other marks you can also say is in the heart, we have a wall that separates the 2 sides that's called the septum. And the septum is responsible for keeping the deoxygenated blood and the oxygenated blood separated. All right, let's move on to part B. An abnormal heart Abnormal heart rhythms can be identified in ECG traces. ECG stands for electrocardiogram. The diagram shows ECG traces of a person with a regular, healthy heart rhythm and a person with an abnormal heart rhythm. All right, we can see those two here. So I've zoomed in so you can see this here. This is the P wave. This is caused by atrial depolarization, which causes the two atria to contract. So that's known as atrial systole. Then we have the QRS complex. This is caused by the depolarization in the ventricles, causing the ventricles to contract. The reason why there's a huge peak like this is because the ventricles are larger than the atria. Lastly, we have the T wave. This is caused by the repolarization of the ventricles, causing the ventricles to relax, also known as ventricular diastole. Okay, let's move on to the next part. Calculate the heart rate in beats per minute of a person with an abnormal heart rate. Give your answer to the nearest whole number. Okay, let's look at the person with abnormal heart rate. So I'm going to zoom in so we can draw this out. Okay, so this part here represents the beginning. This part here represents the end of one heartbeat, and we're just going to calculate how long that one heartbeat is. So that would be 0.84 seconds. So one heartbeat is 0.84 seconds. So this is a ratio question. So in order for me to get from 0.84 seconds to 60 seconds, which is 1 minute, I divide it by 0.84, then times by 60. So what I do to the right, I have to do to the left. 84 and times that by 60. So this is 60 seconds. And the heartbeat would be 71.4 heartbeats. And because they were asking for the nearest whole number, I'm going to write 71. Now they accept a range of 70 to 75. Let's move on to the next part. Part 2, explain how the ECG traces show this abnormal heart rhythm is due to the damage to the septum in the center of the heart between the left and the right sides. So the septum again separates the two sides. All right, so let's scroll back up so we can see. Okay, so the differences between the person with the abnormal heart rhythm and the regular person is the distance between the P wave and the QRS. So you can see the distance here is pretty short, but here the distance between the P wave and the QRS complex is pretty large. That could mean that it might have taken some time for the electrical impulses to go from the atria to the ventricles, which means it has to go through the bundle of His, which is located in the septum. So let's scroll back down. All right, so I wrote that there is a larger gap between the P wave and the QRS complex. And that could be because it takes longer for the electrical impulse to travel down the bundle of His, and that is located in the septum. Another mark you can also say is there is a delay between atrial systole and ventricular systole. All right, part 3. Bradycardia is a heart condition that results in the heart rate that is lower than normal. Atropine is a drug that can be used to treat some forms of bradycardia. Atropine is a competitive inhibitor for acetylcholine. Explain why atropine will lead to an increase in heart rate.
So for your syllabus, you're supposed to understand the autonomic nervous system. You've got the parasympathetic and the sympathetic. So the sympathetic nervous system is responsible for more of those flight or flight responses. So increased heart rate, increased breathing rate, whereas your parasympathetic nervous system kind of slows down, is the opposite of the fight or flight. So decreased heart rate, decreased breathing rate. And in your syllabus, you're supposed to know the two neurotransmitters involved in that. Acetylcholine, which plays a role in the parasympathetic nervous system, and noradrenaline, which plays a role in the sympathetic nervous system. So atropine works as a competitive inhibitor of acetylcholine. Now, if acetylcholine is responsible for decreasing the heart rate, atropine will essentially prevent acetylcholine from binding to the receptors, which is why it would lead to an increase in heart rate. So let's go over the marks. Acetylcholine is released from the parasympathetic neuron. And what happens is that atropine binds to the receptor. So that would be the second mark, and prevents acetylcholine from binding. And this happens at the sinoatrial node. Now, if you recall, your sinoatrial node is the pacemaker of the heart, and that's located in the right atrium. So that's already your 4 marks. Other marks that you can also add is that acetylcholine reduces the electrical impulse frequency, or the frequency of depolarization that occurs in the heart. All right, let's move on to part 4. Describe how exercise leads to the stimulation of the cardiac center in the medulla oblongata. So I've written a summary here to sort of show you what happens when we exercise. So when we exercise, let me just use another marker. When we exercise, we have an increased level of CO2 in our body because we are undergoing more respiration. And what happens is the CO2 dissolves in our blood to form carbonic acid, and that decreases our blood pH. Now, we have chemoreceptors that are in our aorta. We call them aortic chemoreceptors. We also have our carotid chemoreceptors as well. So that decrease in pH is detected by that. And afterwards, impulses along the sympathetic neurons would release noradrenaline as a neurotransmitter to the sinoatrial node. Again, this is the pacemaker of the heart. And then afterwards, that would increase the frequency of depolarization that occurs. So let's go over the marks. So when we exercise, there is an increase in carbon dioxide because of more respiration. So that would be the first mark. The blood pH decreases. That's the second mark. That's detected by chemoreceptors. So I know I only mentioned the carotid artery, but you also have some in the aorta as well. And that would be 2 marks for this. So 1 mark for the chemoreceptors, the other mark for the carotid artery or the aorta. And then electrical impulses are... I forgot to mention acceleratory center in the cardiovascular center. So the acceleratory center increases the heart rate, and then you have the inhibitory center that decreases the heart rate. All right, let's move on to question 9. Phytoplankton are producer organisms in marine ecosystems. Zooplankton are small animals that live in the surface waters of the oceans. This is a food chain for the South Atlantic Ocean. Okay. Part A, comment on the shapes of these 3 ecological pyramids. All right, so I've zoomed out so you can see all 3 pyramids. First, I spoke about the pyramids of biomass, and you can see there's a difference here between the zooplankton and the phytoplankton in November versus December, right? So I made a comment on that. So phytoplankton is higher than zooplankton in November, but not in December. This is probably because the zooplankton consumed the phytoplankton, right? And we can confirm this because that's what it mentions up over here in the food chain. All right, so that would give you 2 marks. Then afterwards, I commented on the energy or pyramid of energy. So I mentioned we can see that as we go up the food chain, the energy increases, and that's because energy is lost. So you can say energy is lost because of respiration, or because some parts of the organism is not eaten, or some of it is not digested and gets egested. I also wrote that it is lost in the form of chemical energy during excretion, or it is ingested, so long as you write out a reason for that. That's another 2 more marks, so this is a total of 4 marks. Another thing you can also say is the pyramid of biomass changes seasonally, so it's different in November than December, but the pyramid of energy is the same. So it's also something you can also say because it's calculated for the whole year. All right, let's go on to part B. The diagram shows the flow of energy through this food chain. Okay, calculate the percentage efficiency. All right. So the first thing I want to do is figure out the energy that is being transferred to the zooplankton. So we know that this is the amount of energy that the phytoplankton has, and this is the amount of energy that is lost. Right, so to find out the amount that's transferred to the zooplankton, we just do this number minus this number. So let's write that down. 6.8 times 10 to the power of 6 minus 6.08 times 10 to the power of 6. And so that gives us 720,000. So 720,000. And then afterwards, to find out the energy transfer percentage efficiency, we do that number divided by this number here. So 720,000 over 6.80 times 10 to the power 6. And because it's a percentage, we times it by 100. So let me write that down in the calculator. And we get 10.58 or 59%. Now, remember to read the question properly. It says to give your answers to the nearest whole number. So that means that is 11%. All right, so that's done. Let's move on to part 2. Common seals and orcas are endotherms. Sardines are ectotherms. Explain why the transfer of energy from sardines to orcas is less efficient than the transfer of energy from zooplanktons to common seals. All right, let's look at the diagram. So it says the energy transfer is less efficient from sardines to orcas. So this is less efficient compared to if you do zooplankton to common seals. Now, the reason why is with the sardines to the orcas, you've got the common seals that's in the middle. And common seals are endotherms, meaning that they need to maintain a constant body temperature, kind of like humans as well. Like we need to make sure that our body temperature is 37 degrees. So because common seals need to maintain that constant body temperature, they also have a high metabolic rate. And they also lose a lot more heat to the environment as well as a result. You could also argue the opposite. So sardines, on the other hand, are ectotherms, meaning that they don't need to maintain a constant body temperature. So they don't need all of that extra energy, or they don't need a high metabolic rate in order for that to happen. Okay, let's write this out. Okay, so you'd have to mention that endotherms need to maintain a constant body temperature. And I wrote that common seals are endotherms. The second mark would be they have high levels of respiration, or you can say they have a high metabolic rate in order to maintain that body temperature, and they also lose a lot more heat as well. So that's 1, 2, 3 marks. Okay. Let's move on to part C. Scientists investigated the impact of global warming on marine productivity. They determined the effect of temperature and light intensity on the net primary productivity of phytoplankton. So they expose it to different light intensities for 3 months, at 3 different temperatures for 1 month. After 1 month, they measured the increase in dry biomass. Okay, state what is meant by the term net primary productivity. So let's go over these terms. You've got GPP, which is the gross primary productivity, and it's basically the rate at which the chemical energy is converted to carbohydrates during photosynthesis. Right, so I've drawn a plant here with the sun. Now, NPP, or net primary productivity, is the GPP minus the rate of respiration. So remember, some of those carbohydrates that is produced by the plant will be used for respiration, which means that NPP sort of refers to the energy that would be stored in the form of biomass, which would then be available for the herbivores or the primary consumers. This is what I wrote. You can also say GPP minus R, and they do accept that as the answer. Part 2. Some effects of global warming include atmospheric carbon dioxide causing an increase in temperature, and that can cause increased cloud cover, and increased ocean temperature would reduce the movement of nutrients such as nitrates from the seabed to the surface water. Scientists concluded that if the use of fossil fuels is not reduced, there would be a fall in populations of orcas. Analyze the data in the table and the information given to discuss this conclusion. Okay. So to get the 6 marks from this question, you have to address at least 6 points from these 3 categories. Number 1 is description. So first, describe the data. What do you see? What are some trends that you see? Right? The second category is explanation. So why is the data like that? And lastly, we want to talk about the validity of the scientist's conclusion, right? Whether or not the use of fossil fuels would reduce the orca population. Okay, so in this question, we're trying to find the link between the use of fossil fuels and the fall of the population of orcas. And we know that if we use fossil fuels or if we burn fossil fuels, that would release carbon dioxide in the atmosphere, which would cause an increase in temperature. And that's because carbon dioxide is a greenhouse gas and it traps more heat into the Earth's atmosphere. Now, when we look at the table, we can see the relationship between the plant biomass and the temperature. So there are 3 temperatures here, 10 degrees, 20 degrees, and 30 degrees. We can see that with higher light intensities, so 20 to 25 arbitrary units, as the temperature increases, the biomass increases, and that's for both of them. So 11, 24, 28, 12, 24, 28, 12, 28, 35. Whereas for lower light intensities, we don't see that trend. We see that it actually peaks at 20 degrees and then goes down, right? So we can see that for all 3 light intensities. The reason why we see a decrease at lower light intensities is because we know that as temperature increases, rate of respiration increases. And that's because the enzymes in the substrate have greater kinetic energy and therefore more successful collisions. So earlier they asked us to state what net primary production was, and that was the gross primary production minus respiration. If there's more respiration that's occurring, which happens when the temperature is greater, then that means there is a decrease in net primary productivity. Therefore, less energy would be available for the primary consumers and the secondary consumers, and therefore the tertiary consumers. So less energy is available for the whole food chain. So from this table, we can conclude that low light intensity and high temperatures would lead to lower net primary production, lower biomass, right? So now let's go back to part 2. So let's go back to the question. So if we look at the second bullet point, we can see that an increase in atmospheric temperature leads to an increase in cloud cover. This is because more water is evaporating and therefore more clouds would form. If there are more clouds, that decreases the light intensity. So again, you have that combination of low light intensity and high temperatures, which leads to a lower net primary productivity or lower NPP. On top of that, it says that increased ocean temperatures reduces the movement of nutrients such as nitrates from the seabed to the surface water. That means that less nitrates would be available for things like protein synthesis for the phytoplankton. So therefore, all these factors, lower light intensity, high temperature, less nutrient availability, would cause the phytoplankton to decrease in population, which would cause, let's go back to the food chain, which should cause the zooplankton to decrease, which should cause the sardines to decrease in population, the seals, and therefore the orcas at the end. Okay, let's go back to the question. Now let's address the scientist's conclusion. It is true that if we do reduce the fossil fuel consumption, then that would lead to less carbon dioxide being released. Therefore, temperatures wouldn't rise as much. You wouldn't have less light intensity, and therefore you wouldn't have a fall in population of orcas. So those are some evidence to support the conclusion. You also have to consider evidence that goes against the scientist's conclusion. For example, the study that they conducted, right, with the phytoplankton, they only did it for 1 month and they only did it once, right? So in order to ensure that your findings are reliable, you need to repeat the experiment or do the experiment longer. There are other things that can cause an increase in greenhouse gases, such as cattle farming. So cattle farming releases both carbon dioxide and methane, and both of them are greenhouse gases. Another thing you can also say is their experiment relies on this food chain, right? It relies on the phytoplankton, the zooplankton eating the phytoplankton, the sardines eating the zooplankton, and so on. But orcas might be part of other food chains as well that does not involve the phytoplankton. So that's another thing to consider. Right, let me write out my answer. So I didn't include all of the points available in the marking scheme, just some that would give you these 6 marks. So combustion of fossil fuels releases carbon dioxide in the atmosphere. That would be a mark for the fossil fuel hypotheses. We know that temperatures would rise. And then I described the data, right? So at higher light intensities, we can see that as the temperature increases, the biomass increases, but at lower light intensities, as the biomass increases, it peaks at 20 degrees, and then afterwards it decreases. And then an explanation for that data. So this is because rate of respiration increases when temperatures are higher. Right, so we have, I'm just going to write this down. So this is F for fossil fuel hypothesis. This is D for description of data, and then you've got explanation over here. Increased temperature leads to increased cloud cover because more water would be evaporating to form these clouds. So that's an explanation. This would lower the light intensity, which means less light is available for photosynthesis. Right, so that would be also an explanation as well. High temperatures and higher ocean temperatures would mean less nitrates available for protein synthesis. That would be another explanation mark. And then putting all these 3 factors together would decrease the net productivity of the phytoplankton. That would mean that less of energy is available for the food chain. And then going back to the conclusion, therefore, by decreasing the fossil fuels, that would lead to less global warming and less cloud cover. So that would be the going back to the scientist's conclusion. Lastly, we also have to talk about the study itself, whether or not the conclusion was valid. Are there any other evidences that goes against the conclusion? So what I wrote here, to ensure that the study is reliable, it is important to repeat the experiment. And scientists also need to consider that orcas might be part of other food chains. All right, let me just zoom out so you can see the whole thing. Okay, so that is the end of paper 2. Let me know if you have any questions about this paper or any other paper that you're doing. Good luck for your exam and happy studying. Bye.